appending additional functionalities to dCas9 to control transcription, edit the epigenome, or image the genome, or mutate the genome. So I think today's a really sort of nicely matched set of talks, I think. Some of what I'm talking about will be a segue into some of the things Mike's talking about, and I'm going to talk about sort of single gene approaches, and then you can imagine scaling a lot of these technologies up to ask additional questions about how the genome controls function of various cell types. So this is the same outline you saw yesterday. We have a lot of slides, so I'm gonna skip through the intro. So initially, as I said, we used dCas9 in bacteria just by targeting this protein to specific sites along the genome. We can inhibit RNA polymerase elongation. This unfortunately does not work so well in human cells, and so we've taken to appending additional domains to more robustly control transcription. And so our goal early on, once we realized that dCas9 alone didn't perturb transcription by much in human cells, was to ask whether we could append domains to either silence transcription or activate transcription to dCas9, so that in essence we have the platform shown on this slide, where we have an RNA-guided DNA binding platform with which we can fuse a variety of effector domains and then easily target these effector domains to arbitrary regions of the genome to enact some function. And so one of the reasons I talked extensively yesterday about the historical perspective of synthetic biology and controlling transcription was we moved very quickly early on because we could take a playbook from what had been done and just repurpose it over to CRISPR. And so I showed you ways that you can silence or activate transcription yesterday using various domains that have been defined over the last 20 or so years. And so we, not being very creative, took a handful of those domains and appended them to dCas9 and asked whether we could control transcription. And so this is one of the earliest experiments that we did, where we took this well-characterized CRAB domain that I had shown you yesterday. This is a scaffold that recruits additional enzymes to change local chromatin marks. And so we appended this domain to dCas9 and transfected or infected reporter cells that expressed a simple GFP reporter with a guide targeting the reporter and this CRAB dCas9 fusion domain, and then just asked a very simple question a few days later whether we could turn GFP expression on. Sort of exactly the same experiment as I showed you in E. coli, but here we're using human cells and we've got some additional effector domains. And so this data is shown on the right, where there's a FACS plot looking at human cells that express either this GFP reporter, and then we can compare those cells to cells that express no GFP, or if we target the GFP reporter with the sgRNA that targets this dCas9 CRAB fusion protein to the reporter, we can see that we can pretty robustly turn transcription off. So these two histograms are largely overlapping, suggesting that there's almost no GFP remaining expressed from this reporter. So this was sort of the first experiment that worked to the point where we were excited about using dCas9 to control transcription in human cells. But our ability to semi-robustly control transcription doesn't really mean that much if it's not a general property. And so we went about testing quite a few guides targeting this GFP reporter just to try to derive rules sort of similarly as we had done in bacteria as to where we needed to position these complexes along a gene to most robustly control transcription. And so you can see, you can test, there's 8 guides up there. Some of them work, not all of them work. We started to learn preliminary rules, and some of those I'll go into in additional detail. But the point of this slide, I think, is to show you that we are actually controlling transcription with these domains. So this is a slide looking at repression of this GFP reporter. If you just target the GFP reporter with dCas9 alone, and here you can see, some of these guides are somewhat active, but you don't really see more than about two-ish fold repression. And if you add this domain, now you start to see, more robust repression. And so we've improved on this over time, but this is our first hint that we actually are targeting these domains functionally to a gene to control transcription, and that this is happening through local changes to the epigenome. So we're not directly editing the epigenome with an enzyme, but we're using a scaffold to recruit endogenous enzymes that edit the epigenome to turn off transcription locally. So what I showed you previously was a reporter. We also early on showed that we could repress endogenous genes, and that we could, similarly to bacteria, repress at least 2 endogenous genes. So this system could be multiplexed. And this was early results. Maybe not, we're not completely turning transcription off, as I'll show you we can later, but these were encouraging that we begin to suspect that we could control expression of a variety of endogenous genes in artificial reporters, either as single genes or as sets of genes, using these artificial transcription factors in human cells. Sort of similar to what I had shown in bacteria, we wanted to know whether we could also use this technology to target promoters to silence transcription, not by targeting gene body, but instead by targeting promoter. And so we were able to show using this well-characterized SV40 promoter driving expression of GFP that we could turn off transcription of the GFP reporter by targeting various sites along the promoter. We also were interested in a more subtle question of whether we could use dCas9 alone, just a protein, in a eukaryotic cell to compete with an endogenous transcription factor for a binding site at a promoter. So you can imagine this is sort of a different question where we were sort of very, in a simple way, probing whether we could map regulatory regions across the genome by competing to proteins for a specific binding site. And you can imagine this would have a variety of applications in mapping enhancers or promoters. But the first experiment we did to test this was we didn't use a completely endogenous system, but we had a reporter that had TET binding sites that drive venous expression, and we constitutively expressed a TET activator protein that induces venous expression. This is in yeast, actually, so you can see that when you express the this RTTA or this TET activator protein, you induce venous expression. And then if you co-express dCas9 targeting these TET sites, we see that we can turn gene expression off. So this is illustrated here. We don't go all the way back to baseline, but it clearly illustrates that we can use dCas9 in a eukaryote to compete for a transcription factor binding site and control gene expression, sort of functionally mapping sites in an artificial way, functionally mapping sites along a promoter which are required for controlling gene expression. And so this is, I think, sort of an underexplored use of dCas9, but it has a lot of potential for mapping regulatory regions, sort of in a very fun way. So our ability to control transcription is only useful if it's specific. So if you have, it doesn't matter how robust your ability to control transcription is, if you're deregulating many genes across the genome, that's not going to be a very useful tool for understanding how biological systems work. And so I'm not presenting these experiments in temporal order, but after we saw that we could use these dCas9 crab fusions to edit the epigenome and control transcription, we immediately went and did an RNA sequencing experiment to see whether our ability to control transcription of these reporters was specific. And so I like this experiment for a couple of reasons. One is it gave us the answer we want. So that data is shown here. This is the transcriptomes of 2 populations of cells, one of which expresses a negative control and the other expresses a sgRNA targeting this GFP reporter. So when we express the sgRNA targeting GFP, we guide the dCas9 crab fusion protein to the GFP reporter and turn transcription off. You see that GFP expression goes down. Very satisfyingly, though, we saw that really the expression of no other genes change. So this implies that we can both robustly and very specifically control transcription. When we saw these results, this is really where we got excited about using this as a means of controlling transcription in human cells. The reason this experiment works sort of well, and the reason we did it, was that you can imagine it can be hard to test specificity of your specificity for modulating transcription for endogenous genes. If you overexpress or knock down an endogenous gene, that's going to have primary effects on the gene you're targeting, but could also have a variety of indirect effects. You can imagine imagine if you're knocking out an enzyme that's required for some metabolic function, you're going to knock that enzyme down, but then over time you're going to accumulate secondary transcriptional changes. And so one of the ways that we like to measure specificity is repression or activation of proteins that have no functional consequence on the cell. And that allows you to really measure your direct effect on transcription and not convolute this with other effects. So these are additional control RNA experiments that show that expression of a guide alone or the Crab fusion alone don't really have much of an effect on cells. So these aren't like grossly toxic to the cell. And this is an important point for thinking about, applying these to a variety of models. You wouldn't want to have some gross underlying toxicity if you're trying to study some stress response or developmental response. Similar to what we showed in bacteria, we showed that our ability to turn off transcription in human cells is controllable. So we can create inducible systems where we can induce the expression of our CRISPR constructs to turn off transcription acutely, and then we can withdraw these inducers. In this case, we're using doxycycline. The promoters go off, and the artificial transcription factor goes away, and the genes go back on. So that's shown more or less here, where we've done an experiment where we knocked down these two genes. We then wash out doxycycline, so DCAS9, the CRISPR expression goes off, and you can see transcription comes back on. So with delayed kinetics relative to what I showed you yesterday for bacteria, but nonetheless suggests that our ability to control transcription is inducible and reversible, and the changes that we make to editing the epigenome are not inducing permanent silencing of a gene, but rather are locally changing chromatin in a way that is fully reversible. Which ones? Yes. No. So you're talking about the anti-CRISPRs. Yeah. No. So we've used more sophisticated schemes to control activity where we have small molecule dimerization systems where you can. What was shown? One of the things that showed me yesterday was these split Cas9 systems where you're not controlling transcription, you're controlling protein dimerization. Using a small molecule, we have done that. We have not used an anti-CRISPR to control our DCAS9 constructs. You definitely should be able to, but we're doing those experiments, but they haven't been. We've been a little short-handed recently, and so we haven't gotten around to them. The anti-CRISPRs are relatively new, so that is really have only been applied in human cells over the last 6 months or so successfully, and we just haven't had strong biological motivation for testing that. I mean, I think it was shown to work with Cas9, and so once that worked, it's very likely to work with DCAS9. But definitely an additional way of control would be exciting.
Except that anti-CRISPR genes have been complex, so they might not be effective in preventing transcription.
That's true. We don't actually know. You're right. Although for the one the anti-CRISPRs all, the first three characterized, all have different mechanisms for inhibiting cascade. So even if the one that was shown yesterday doesn't work for DCAS9, there's some rationale to think that we could find one that would work. But we'll see. We haven't tried. We've appended it to both. I think. No, I actually like the C-terminal fusions better. I think they're a bit more stable. The N-terminal fusions, you can see the protein is quite unstable. So when you wash out doxycycline here at day 4, by day 5, the protein by Western blot is almost completely gone. The half-life is quite short in human cells. That's a good thing for an inducible system because it makes it less leaky. It means to get in our hands, you need a reasonably strong promoter to get full activity of these N-terminal fusions, where you can use a weaker promoter for the C-terminal fusions just because the protein is more stable. So the bottom plots also show that you can use this to study essential genes. So one of the things that is a little bit tricky to study using wild-type Cas9 is an essential gene. If you make an edit to the genome to knock out an essential gene, that's a bit difficult to recover a dead cell. With CRISPR-I, we can use these inducible versions of CRISPR-I to control transcription, and that way we can set up experiments and then knock down an essential gene when we want, and then carry out analysis. So here below, this is the same inducible system where before we're looking at knocking down these essential genes over the course of several days. If we don't add doxycycline to turn on expression of the CRISPR-I construct, we can see that these sgRNAs targeting essential genes have no effect on cell proliferation, so the cells proliferate like wild-type cells. If you then knock down these variety of essential genes, you see that the cells where you've knocked down the essential gene deplete very robustly relative to their wild-type counterparts. And then lastly, we've shown that you can use it to control expression of both protein coding genes and non-coding genes. This is a little bit later work than what I showed earlier, where we're knocking down a variety of endogenous genes. And you can see that our ability to control transcription has gotten more robust over time as we've derived algorithms that are more highly enriched for highly active guides. So this is qPCR data on the top and the bottom, and it's a log 10 plot. So you can see we're really robustly controlling transcription for many of these genes, both protein coding and non-coding, where we're getting like 99.9% repression. And if you look by, for example, FISH down here for the exist RNA, which coats the inactivated X chromosome, you can see that there is like really no signal left at all for these cells. So we're completely turning off transcription for this endogenous non-coding RNA. So all that work was done in cancer cells, a chronic myeloid leukemia cell line called K562. Not all of it, but most of it. Some was done in HEC293s and HeLAs, but really like standard sort of very robust and easy to use cancer cell lines that may or may not model many types of biology. And so we were curious whether we could expand our ability to control transcription out of cancer cells into more developmental models or other models. And so together with Bruce Conklin's lab, who's in the Gladstone at UCSF, we decided to ask whether we could control transcription in human iPS cells. And so what we did was, rather than all the previous work I showed was either transient transfection or lentiviral delivery. In this case, we've gone to knocking our constructs into a safe harbor locus within the cells, so we can control their expression very stably over time in iPS cells. And then we use the same inducible system that I showed on the previous slides to ask how efficient is our ability to control transcription? Is this tunable? Is it reversible? Can we multiplex this? We continue to ask many of the same questions. And so this is a series of pictures comparing the ability to genes not on here. They're targeting what is this? So this is actually just a schematic to show that the system works, that you can knock these constructs into the genome of an iPS cell, and that we can control expression of either Cas9 or dCas9 within a regulatable system. So in both cases, we've targeted either our CRISPR-I constructs or a Cas9 nuclease to the AAVS1 locus. We can use a TALEN to make a double-strand break, which increases homologous recombination, and then we use a long double-stranded linear donor to knock in all of this business into these AAVS1 loci. And so what we've knocked in is both a constitutive promoter driving the RTTA that in the presence of doxycycline will activate expression of a CRISPR-I construct or a Cas9 nuclease. And you can see that in the absence of doxycycline, the CRISPR-I construct or Cas9 are not expressed. If you add doxycycline, you get homogeneous expression across the population of cells. So then the question is this functional? And so Bruce's lab targeted a variety of genes, but shown here is targeting NANOG with either CRISPR-I or CRISPR-N. And we're able to show that here in iPS cells we can very robustly control expression of this gene NANOG. So NANOG here will be stained in green, and you can see there's no green there, so that's pretty good control of gene transcription. If you target NANOG with CRISPR-N, you see more heterogeneity. So some cells will be completely knocked out for NANOG, other cells will be heterozygous and still express some NANOG. Both tools nulls are great. They just give you different outputs. Here, you're not making nulls. You're turning transcription down. Here, you make true nulls, but you also have heterogeneity in protein expression across the population. And Bruce's lab showed that this system in general, as in cancer models, works quite robustly to control transcription of a variety of types of endogenous genes. This is just qPCR data. But overall was a nice proof of principle set of experiments showing that we could turn off transcription in iPS cells in an inducible way. And so you can imagine this is especially important for studying differentiation, where you might want to grow your iPS cells up, infect in a guide, induce some differentiation protocol, for example, that guides you down a neuronal lineage or a cardiac lineage, and then at a specific time point during that differentiation process, turn off a gene and ask how is that gene required for neural differentiation or cardiac differentiation. And so that's all possible with either of these systems, really, in most ways, but definitely we're able to control transcription in iPS cells. One of the things we noticed really early on that I like to reemphasize in this workshop is that in many settings in human cells, expression of an sgRNA is the limiting factor for a lot of types of experiments, whether it's imaging or, in many cases, editing, if you're not using RNPs, or